Good. I think we are live, so can anyone give me a thumbs up if you can hear me and if everything is fine? Can you hear me?
I can hear you.
Yes.
This is Chris.
Yes. Yeah, okay. I think there's a slight delay. Perfect. Good, great.
All right.
Perfect. So let's start then. I'm very excited to start this new series. And so just before starting, just let me give a few technical, make a few technical remarks. So first of all, I mean, this is going to be there. I mean, it's a series that's going to be every Tuesday at this same time. And I would like to thank already Team Vogel and that actually makes this possible and is coordinating this effort. And also Camilla Bellone and Laura Pancetta that are helping me in putting this impressive lineup of speakers. So with that, I'd like to introduce our first speaker. So Chris Walsh earned a bachelor degree in chemistry from Bucknell University in Pennsylvania. He then went on to graduate school at the University of Chicago, where he obtained his MD and PhD in 83 and 85, respectively. I think actually the other way around. He completed a postdoctoral fellowship in the laboratory of Konstanz Chepko at Harvard University, where he was also appointed as professor of genetics in 1993, and where he remained ever since. So Chris Walsh studies disorders of cortical development, such as lissencephaly, epilepsy, and cortical dysplasia. And his lab uses human genetics as an entry point to understand basic and functional mechanism of those disorders. I know, I mean, Chris is a Howard Hughes investigator as well as a member of the Academy of Sciences. Needless to say, he has won several awards. But very importantly, also know he's a great mentor, and in fact, there are many great scientists coming out of his lab. Again, I am very excited about having you giving this talk, Chris, and I thank you again for this. So everyone can post questions in the tool down there to ask question tool, and we can ask a question at the very end. So with no further ado, I guess the worldwide neuro is yours, please.
Hi, everybody. Thanks very much, Gaia, for that very generous introduction and for the invitation to talk today. I'm coming to you from our dining room at our house. So I'll apologize in advance if the doorbell rings because the FedEx guy is bringing our food delivery or if somebody uses the phone, but it's great to have a chance to try to keep doing science under these difficult times. Also, I can't see anybody. All I see is my screen, so it's a little bit of a bizarre thing to have no visual feedback, so I'll try to do the best I can. So I'll be telling you about some recent work from our lab. Most of what I'll be describing is unpublished, although much of it is available on bioRxiv. And so our lab's been interested in developmental disorders of the human cerebral cortex for now 2 decades. I just have always thought of these as not only important causes of human illness, but also an incredible way to do science. We see just incredible diversity of these conditions. This just shows, looks like I don't have a pointer, so I'll have to describe things. The MRI scans come from individuals with 8 different Mendelian disorders of cortical development that are recessive or X-linked or dominant in some cases. There's an incredible diversity of these disorders. We're still describing new ones every week. They invariably seem to uncover developmental mechanisms that we hadn't really thought very much about based on worm or fly studies. Some of the genes enlighten us about human-specific features, And so I was very inspired by this quote from Sidney Brenner in his Nobel Prize acceptance speech that he proposed at that time a project he called Humanity's Genes, where really this tremendous diversity and essentially saturation mutagenesis of humans for mutations in any gene that's essential for development could be used to systematically discover genes and mechanisms that are required for the process, in this case, the development of the human brain. And then we've used animals as more of a way of validating them. So what I'll tell you about today is a part of the interests of our lab, focusing on mosaic mutations, those that are present in some neurons of the brain, but not all of the neurons of the brain. So emphasizing this fact that even though we have one brain, we have many, many genomes, because every neuron has a unique genome. I'll talk about somatic mosaic causes of focal epileptic disorders. And that's mostly published, so I'll cover that briefly, mostly to introduce the paradigm. I'll talk about somatic mosaic causes of autism spectrum disorders, some published and some unpublished work. And then somatic mosaic mutations as cell lineage marks, just to use the natural occurrence of mosaic mutations to study the sequence of development of the human brain at postmortem. And then finally, I'll talk a little bit about areas that we know less about, how these disease-associated mosaic mutations fit into the normal cell lineage. Are there mosaic disorders of specific cell types in the human cortex? Or can mosaic mutations be used as experiments of nature that might illuminate critical mechanisms of disease? And that's an area that I think at this point is as yet not resolved. So we started working on mosaic mutations in epilepsy due to the work of an amazing fellow, Anne Padori, who is a child neurologist who's had her own lab now for several years. And she wanted to understand a condition called hemimegalencephaly, where half of the brain is grossly enlarged. Can people see my pointer when I move it? Can people see this arrow? Anybody there?
Yes, actually,
Yes? Okay.
You can see it.
Marvelous. Great. And so this is a patient with hemimegalencephaly, in this case affecting the right hemisphere. And that right hemisphere was overgrown, as you can see, particularly here in the axial view. It's larger than the normal hemisphere. It's grossly disorganized. You don't see good differentiation of gray matter and white matter. And that patient had intractable epilepsy starting on the day of birth, along with left-sided weakness. And so that entire right hemisphere was removed surgically. As you can see here, where there's nothing but white, that's cerebrospinal fluid where the cortex used to be. And that was life-saving for this young boy. He had no seizures for 6 years. He learned to walk and developed fluent speech, although he was born with left-sided weakness because the right hemisphere was not functional. And he's still had some persistent left-sided weakness. Here's a picture of him. His name is Dante. I use his name and picture with the permission of his parents. As I say, it's been life-saving for him. He reads at grade level. He walks. He talks fluently. And he's by no means normal, but has had a remarkable life considering his congenital disorder. So studying a couple of other cases, here's another case, in this case with overgrowth of the left hemisphere, and it looks like my pointer doesn't work on this slide. And this patient and another patient, they both had a gain of the entire long arm of chromosome 1, as shown by this old genotyping array. You can see darker pink for the area inside the circle, and that's the entire long arm of chromosome 1. And you can see that that's present at a copy number between 2 and 3, but then when you sort single cells from the brain, you can find single cells that either carry the copy number increase as shown in panel B, or cells that have normal copy number for chromosome 1 as shown in the panels below panel B, where there's normal copy number on the two sides of that vertical gray bar. And we found that this particular CNV is a clonal CNV that arose during the development of the brain and was present in about 20% of neurons and about 20% of glial cells, but undetectable in the blood of the same individual. And that long arm of chromosome 1 contains a gene called AKT3. In fact, Dante's mutation, the boy I showed you, he had a point mutation of AKT3 as shown here in this slide. You can see a small peak under the black arrow, which is again a mosaic mutation present in some cells and not others, and absent from the leukocytes in the row below that, where there's none of that smaller reddish peak. And this is a particular activating mutation in the gene AKT3, which is a known oncogene. And this is a known oncogenic mutation in AKT1, which causes constitutive activation of the AKT3 kinase, and which is known then to stimulate overgrowth. So this point mutation then is responsible for this condition, and again arose during the development of the brain and is only present in the abnormal tissue and not detectable in the blood of the same patient. And we now know through work of Anne and Alyssa DeGamma, a former PhD student, that these gain-of-function mTOR pathway mutations cause a range of visible malformations. They can cause this large hemimegalencephaly, or they can cause a smaller condition called focal cortical dysplasia, as shown in the MRI that's second from the left there, panel B, and that a range of mutations in the whole mTOR pathway, the PI3 kinase gene, PIK3CA or PIK3R2 or AKT3, which is upstream of mTOR, or most commonly in mTOR itself, can cause either these small lesions, the focal cortical dysplasia, when the mutation is present in a tiny fraction of cells within the lesion, or if they occur earlier, they can cause the hemimegalencephaly. And our work was simultaneous with work from many other labs, notably Stephanie Baulach in Paris, and Bill Dobbins in Seattle, Joe Gleason in San Diego, and Jung-Ho Lee when he was in Joe's lab, and since he's had his own lab now in South Korea for many years. And so these mutations can occur then, these mosaic mutations can occur early on before gastrulation, or they can occur later and later in cortical neurogenesis, where they occur in smaller and smaller pieces of tissue and smaller fraction of all of the cells within that piece of tissue. So here's an example where the same mTOR mutation can cause two different malformations depending on when and where the mutation occurs. So in this case, we have three patients. They all have a T1977K substitution in the mTOR gene itself. And the first case on the left has a parieto-occipital focal cortical dysplasia, and when that dysplasia was removed surgically, the mutation was detected in 3% of the cells within that lesion, but not in the saliva of that same patient. You can see the middle patient had a focal cortical dysplasia in a different location in the frontal lobe, and again had the same mutation, again at about 3% of the cells in the lesion. And then the right patient had hemimegalencephaly, had the same mutation, now present in 15 or 20% of the cells within the larger lesion when it was removed at surgery, but not in the blood of that patient. So these mosaic cancer-like somatic mutations can occur really at any time from the first cell division to the last. And the disease that they cause then is determined not only by the mutation, not only by the allele, but also by the timing of when it occurs. So mosaic mutations, we now understand, can occur at any time during the development of the body. They can occur early, in which case they'll be present in a fraction of cells in all the tissues of the body. They can occur a little later, in which case they're present in a fraction of cells, but limited to the brain. Or they can occur in progressively later stages, where they might be in one hemisphere and not the other hemisphere, or perhaps in a small portion of just one hemisphere. We also know that they can actually be non-clonal and can be limited to a single non-dividing neuron, and we've shown that in other studies, but I'm not going to talk about that for this talk, where I'll be focusing on disorders of neurodevelopment. So our next question was, since mosaic mutations are very important as a cause of epilepsy, where our lab had also been long interested in autism spectrum disorders, we also wondered whether there might be a subset of disorders of autistic disorders that are also caused by mosaic mutations. Now, autism spectrum disorders are typically de novo. They can be caused by de novo copy number variants, which are deletions or duplications. They can be caused by de novo coding region point mutations. Excuse me while I move to a different room to try to get a little bit more quiet. And so since they're frequently de novo, we wondered whether some of them then might occur mosaically and be present, again, like the epilepsy mutations, in some cells in the body, but not all of the cells of the body. So in the case of de novo copy number variants, we know that they occur in 5 or 10% of autism cases. And so, again, we wanted to ask whether mosaic copy number variants might also contribute to autism risk. And this is work done by Max Sherman, who's a graduate student in Po Ru Lo's lab. So we collaborated with Max and Po Ru, and Rachel Roden and Caroline Diaz from our lab were involved in that. So Max analyzed blood DNA from 12,000 autistic probands and 5,000 unaffected siblings in the Simons simplex cohort, as well as the Safari Spark collection. He used a newly developed haplotype phasing method developed by Po Ru when he was a postdoc with Alcus Price, which detects mosaic CNVs even at a very low level of mosaicism when they're large. So what he found was that mosaic copy number variants contribute to a small but a consistent proportion of cases, about 10-fold less common than the germline de novo CNVs, so about a quarter or a half of a percent, or a quarter to a third of a percent of cases. So you see there's a large excess of these mosaic copy number variants in probands compared to siblings, both in the combined data sets as well as in Spark and SSC individually. This shows the size of these events. And you see that the proband sizes are consistently larger. It's particularly, for some reason, these large mosaic CNVs that involve large pieces of chromosomes that seem to contribute to risk. These copy number variants that are mosaic in autism actually are also larger than the CNVs that are present and causative in the germline state in autism spectrum disorders. Here are some examples of the amazing sorts of cases. Here's a case where there's a mosaic distal deletion of chromosome 18, and this deletes a gene TCF2, which is known to cause a very severe developmental disorder. And this patient has many of the hallmarks of that of loss of TCF4. And this is a fairly early mosaic event, so it's present in about a quarter of the cells. And you see the distal deletion by the area of low copy number shown in red. Here's another interesting case where a patient actually had a maternally inherited start loss mutation at the initiator codon of neurexin-1 beta in 100% of the cells, but then actually on the paternal chromosome had a mosaic deletion present in 8 or 10% of the cells, which either of these events by themselves are probably not causative, but because of this, there's actually compound heterozygous loss of part of both alleles of neurexin-1 in a fraction of the cells in this patient's brain. A brain we assume based on the fact that it's present in the blood. Or here's another case where there's actually a copy number neutral loss of heterozygosity, so essentially paternal uniparental disomy of chromosome 11p. You see there's a loss of one of the two alleles is shown in the red shading under maternal allele fraction, but in the total genotyping intensity, you see that there's no gain or loss of DNA. So this is a swap of the maternal for the paternal allele, but this particular region, this 11p15.5 imprinted region, in fact, causes a syndrome called Silva-Russell syndrome, which when you have, unless you have an active maternal copy. And so this is a loss of the maternal copy in a third of the cells, causing, again, an autistic syndrome. So what's interesting about these mosaic events is that they are never the events that we think of as the common recurrent autism events. There are certain recurrent autism CNVs that affect chromosome 16p11, chromosome 22q11, chromosome 1q21, and none of these are present in the mosaic state in any of our autism patients, as you can see under panel B, where the mosaic CNVs are completely absent in the autism cohorts. In fact, these recurrent events are common as germline events, but they're absent in the mosaic state. In fact, they're more common in normal people in the mosaic state. You can see under UK Biobank, there's actually a couple of cases of mosaic CNVs of these regions. And in the UK Biobank, in fact, these mosaic events at known autism loci don't seem to predispose to any of the phenotypes that these same CNV events contribute to when they're in the germline state. And so mosaic mutations in autism don't simply cause, in all cases, a milder version of the germline disorder. They can do, but they don't always. And in some cases, it seems like these recurrent events have to be present in the germline in order to create risk for autism, whereas the mosaic CNVs have to be of a different sort, typically much larger. We've also looked at mosaic point mutations in autism spectrum disorder. And this is actually a summary of published work from a couple of years ago by Elaine Lim, a former postdoc, who's actually just starting her own lab at UMass Worcester next month. And she looked in trio sequencing from almost 6,000 autism trios from the Autism Sequencing Consortium. And she found that most de novo mutations are, as expected, heterozygous and therefore have an allele fraction of 50%, 0.5, which is what you see in the large peak in the blue histogram and the dotted red line, but that a certain fraction of patients have de novo events which are only present at a lower allele fraction of about 25%, and these represent potential mosaic events that occur not in the sperm or the egg, but occur in the first cell division or the second cell division, and so are present only in a fraction of cells. So these would be early somatic events that occur before gastrulation, which is why they would be shared in the blood and presumably in the brain as well. And this is what they look like in next generation sequencing traces. You can see a heterozygous germline event in the right in red, where it's present in half of the DNA reads because it's heterozygous in all of the cells. And the mosaic event has, in this case, a variant allele frequency of 4%, which means it would be, again, heterozygous in about 8% of the cells. So the mosaic fraction is about double the allele frequency. And I'll use both of these terms to describe somatic events. So Elaine found that there was an excess of damaging missense mutations in genes expressed prenatally. Some of these occur in known autism genes, others in novel genes. These mosaic patients more often have preserved IQ than patients with germline mutations. And then there's, again, important work from other labs, Jonathan Pevsner and Liping Wei in Beijing, and also Brian O'Rourke, that found similar events and suggested that mosaic mutations may contribute to up to 5% of autism cases, although we really don't know exactly what fraction of autism cases are due to these mosaic point mutations yet. And this may be a mechanism then for high-functioning autism. So in recent work, which is still under review, Rachel Roden, a recently graduated student, and Yanmei Do, a postdoctoral fellow in Peter Park's lab, have been looking at a dense 200x whole genome sequencing of bulk DNA from postmortem brain in 59 autism cases and 15 controls to get a sense of the overall landscape of somatic mutation in both the normal and the autistic brain, as well as to see whether there might be differences between the two. This approach has very good sensitivity for detecting mosaic SNVs with allele frequencies of greater than about 5%, so in other words, present in 10% of the cells, but is actually insensitive even below that. And even at that relatively high rate of mosaicism, we detect an average of 25 or 30 mosaic mutations per brain, ranging anywhere from 2 to over 30. They occur throughout the genome. These validate at a very high rate. Yanmei developed a very good calling algorithm for detecting these things very accurately with a very high validation rate. We also see occasional indels, small insertions or deletions that are clonal in this brain DNA. And again, this shows what they look like in next generation sequencing traces. So we can then estimate what fraction of us, normal individuals, have a clonal damaging point mutation in some important gene in a certain fraction of cells. And we estimate that a typical person has a damaging clonal event in 2% or more of the cells of our brain. And we assess this two ways. First of all, by estimating the number of point mutations per cell cycle. And we can measure that there are 3 or 4 point mutations in the first cell cycle, and then 2 to 3 in each subsequent cell cycle. And that's an average of almost a half of a damaging SNV in the first 4 cell cycles. If you extrapolate that to the 5th cell cycle, again, a typical person will have a damaging mosaic SNV in 2% or more of their cells. We did it a second way. We did high depth sequencing from multiple brain areas. So in a couple of brains, we also sequenced DNA from area 17 and area 18. We then looked for mosaic mutations that were detectable in frontal lobe, but undetectable in occipital lobe. And this allowed us then to estimate how our sensitivity to detect regional SNVs as opposed to ones that occurred at an early stage and were present throughout the brain. We estimate there are about 80 mosaic mutations over the first 5 cell divisions alone, compared to the 100 or so de novo germline mutations. So we think that these mosaic mutations during the first couple of cell divisions are an important source of both variability as well as potentially disease in some cases. So mosaic SNVs, again, likely to contribute to some small fraction of autism, but also potentially functional heterogeneity between neurotypical individuals. Autism brains, when compared to normals, actually show surprisingly an excess of mosaic mutations in sequences that have marks of brain enhancers. I say surprisingly because we have such a small sample size, only 59 autism cases and 15 controls. It's really surprising that we would have the power to detect an excess of anything. And yet, mosaic mutations in autism brain show enrichment versus control in sequences that have active enhancer marks in multiple neural tissues. You can see here there are 29 of these mosaic enhancer mutations out of 59 autism brains and only 1 in the 15 control brains. The fact that these might contribute to risk is further supported by the fact that genes within 50 kilobases of these mosaic enhancer mutations in the autistic brains are also enriched for brain-specific expression, as you can see in the histogram here. And then the rest of the figure on the right side of the slide just illustrates one of these mosaic enhancer mutations in an in vitro assay and shows that, in fact, it actually does impair the enhancer activity of that particular sequence. So we showed that for some of these 20 or so enhancer mutations, there are several of them we can show that they actually impair enhancer function. And a lot of them alter transcription factor binding sequences. As you can see in the upper right-hand corner under panel D, there are predicted transcription factor binding sequences that are altered by the mutation. So to summarize this part of the talk, somatic mosaic mutations cause brain diseases. They can contribute to occasional cases of autism spectrum disorders by several mechanisms. Mosaic CNVs, accounting for perhaps 1/4 or 1/3% of cases. Mosaic SNVs, probably accounting for a couple of percent of cases. And then mosaic non-coding mutations occurring for overall percent of disease that we haven't yet determined, but probably also in the range of a couple of percent. Mosaic mutations in neural enhancers are especially common in autism. They'd be expected, interestingly, to affect the brain-specific expression or the function of a gene without necessarily affecting the gene's function in other tissues, because you're not knocking out the whole gene. You're just knocking out the control of the expression of that gene in the brain. And so you can think of these mosaic mutations as sort of a way of nature performing these experiments to impair certain genes in the brain and in a fraction of cells in the body. And they would sometimes go through the germline if they're occurred fairly early, but not always. Finally, mutations in a small fraction of cells in just a little region of the brain can, in some cases, cause disease. I remind you that mutant cells in 1 or 10% of cells in a small part of the cortex can cause focal epilepsy. So the second half of the talk, I'll talk about using these mosaic mutations for mapping cell lineage in the cortex. So what I've told you is that there are 2 to 3 point mutations per cell division, as well as occasional mosaic CNV events, transposon mobilization, microsatellite mutation. And so that by the time neurons are born, a typical prefrontal cortex or PFC neuron actually has hundreds of unique SNVs not present in the parent or the zygote, and then a different neuron next to it will have a different set of mutations reflecting its distinct proliferative history. So that each neuron then in principle has a barcode in its genome reflecting its proliferative origin. And so that the normal brain, and as I say, normal brains typically have a damaging mutation in perhaps 2% of the cells, and some of these cause diseases, but in fact we ought to be able to read out these mosaic SNVs as a permanent forensic record of the cell lineage of any given neuron. We showed in work published a couple of years ago that you can reconstruct a little bit of this lineage. Again, it seems my pointer is not working, but this shows genotyping of about 100 some odd cells. out of about 200 that were genotyped. And some of these cells were sequenced for their whole genome. Those are shown by the shaded boxes around the periphery. And you can see that the A1 cells, there are about 30 of them, they all share a particular mosaic SNV called, which I just referred to as mutation A1. So we know that those cells are all part of the same clade or the part of the same lineage with respect to one another. And then a different set of cells labeled B, they all share a light green mutation, and then some of them share later mutations that were present in a fraction of the cells, but not all of the cells. And you see that the C clade is a larger clade still, and again has a whole nested series of mutations, which are present in progressively smaller fraction of cells. And so that this illustrates how you can reconstruct lineage from these, in this case, single neurons. And this is work from two former postdocs, Mike Lodato and Molly Woodworth, and then Sarah Bazzotto and Javier Gans have added to this particular figure. So we can actually assemble, at that time, we could assemble more than half of the cells into these 5 lineage trees or clades. And this shows then you can also supplement this by taking samples of bulk DNA from regions of the cortex. And that's what's shown in these drawings of a human brain with these little dots, which represent sites where we sampled the DNA, and then just determined whether a particular mosaic mutations are present or absent. And so in this case, we can organize the mutations in that C clade into those which occurred late and which are present in a small region, as shown in the right side, right drawing, where you see just a little bit of blue shading in the frontal lobe, which is where the mutation was originally identified. And then you can see to the left of that an earlier mutation present in a larger region and in a higher fraction of cells. And then mutations on the right side, the left side of that row are present throughout the whole brain, as shown here by the arrow. In this case, this was a hemispheric mutation present in one half of the brain, but not the other. So recently, Sarah Bazzotto and Jan May and Javier have been analyzing this data in tremendous granular detail to try to really define as much as we can about the lineage of 2 particular individuals about which we have a lot of deep sequence information. And so they've been able to define, using multiple mutations, essentially the first 8 cell stage of the embryo. They can define mutations that define the first cell division, as shown here, and then the second cell division in one branch, and so on. They can see, for example, that these 8 different cells contribute differently to different tissues, as shown by the shading in panel B, where the gray matter and the white matter of the brain tend to have a relatively similar contribution of the different cell types, but then the liver has a much larger contribution of the orange clades than of the blue clades. We can also see that cells contribute asymmetrically to the embryo, because if you look at the red circles, it shows the allele frequency of the mutations that are sequential. You see the first one is present at allele frequency of about 0.24, but the next mutation is present in about 0.15 and then 0.13 and 0.12. The allele frequency should be, if the cells were dividing symmetrically and mixing completely, then the allele frequency should be reduced by 1/2 with each subsequent cell division. But that's not the case. In fact, it reduces by less than 1/2, suggesting that the segregation is asymmetrical. So I'd like to note, can these mutations be used to compare cell lineage between human and mouse? And this just shows one of the major features of cell lineage that we know from animal studies is that excitatory neurons generally form focal clustered patterns. And we've known this again for 20 years or more, but it's been very beautifully illustrated recently by people like Songhai Shi and Simon Hippenmeyer and others who have used the MADAM method to illustrate these cylindrical clones in the cerebral cortex of the mouse. But we also know that non-pyramidal neurons show a totally different origin in the basal forebrain, the ganglionic eminences, and then disperse so that a single clone of inhibitory neurons is shown by a former graduate student of mine, Chris Reed, using a retrovirally barcoded library, that a single clone of inhibitory neurons like the blue or the green clones in this ferret cortex can disperse from one end of the cortex to another. So Defining lineage of neuronal cell types requires simultaneously capturing both lineage marks as well as markers of cell type in single cells. And this is a huge technical challenge, which I think we're still evolving the best method to solve. Analyzing DNA somatic mutations and RNA markers in a single cell experiment is very challenging. The DNA markers occur throughout the genome, a modest bias for open chromatin and in exons. Few of these DNA mutations occur in exons that would be available to RNA sequencing. And also single-cell RNA sequence provides very light biased coverage, so it's very hard to call mutations anyway. And other methods to identify cell types also don't cover most of the genome. So again, Sarah and Javier and Yanmei have been seeing what we can see of these brains where we have very good information about mosaic variants. Can we pick them up in RNA-seq and ATAC-seq? And this shows an unpublished experiment where they've aligned RNA-seq and ATAC-seq embedding from the same brain to separate various cell types, and then looked at known mutations of these 4 different clades, numbered C1 to C8, to see whether we can see them in different cell types. And in this case, you can see that, in fact, as in panel E, you can see that some of these clades are much more common than others. You can see that clade 8 is particularly common in many different cell types, whereas clade 7 is rarely seen. So that different cell types in the brain are contributed by different early progenitors. And then you can see this differential representation. For example, in this case, microglia in panel F tend to be derived to a large extent from clade C3, whereas other clades actually are very rarely seen represented in microglia. So we can see then that distinct early cells of the embryo definitely contribute differentially to cell types of the cortex. But for looking at this comparison of cell type and cell lineage, we needed a method that would give us a higher yield from many, many different cells, and that would allow us to analyze sequential marks in the same cell. And so for this, Peng Li and August Yue Huang, 2 postdocs, developed a method we called paired-seq, parallel analysis of RNA and DNA following deep sequencing. And so this then sorts single nuclei and then uses a targeted approach using specific PCR primers for either the DNA variants or for RNA for about 30 different genes that are markers of the major cell types in the cortex. And then does both of this at the same time, and using a fluidine biomark apparatus, then maps the cells back onto cell types, onto the Teasney plot. And this is a paper that's available on bioRxiv as well. So to do this, we first generated our own single-cell RNA-seq data set using 10x genomics from the prefrontal cortex, which is where we discovered our DNA variants of this normal postmortem brain. And that shows a subdivision of the neurons into excitatory and inhibitory cells, and then subtypes of excitatory cells in the upper layers, the middle layers, or the lower layers. We also took advantage of a recently published data set. We collaborated with the Allen Brain Atlas and used their data set, which was derived from the middle temporal gyrus. So a different part of the cortex from a different individual, but both association cortices. And we found that, in fact, our subdivision of cell types very closely matched theirs, as you can see from this panel on the right, where the 2 different data sets are compared. And we see the same major principal cell types. And then we organize the DNA mutations based on their presence and absence in single cells, and also the mosaic fraction, or the allele frequency of the mutation in bulk DNA. And you can see here that progress under the dotted line on the left, that dotted circle on the left, that mutations that occur within the same clade but at later stages have a lower mosaic fraction in bulk DNA. And then you can see on the right side that mutations C4, C5, C6 occur at progressively later stages are also present in a smaller fraction of cells among the cells that were assayed using paired-seq. And you can also see that, in fact, in the case of those mutations C4, 5, 6, they become progressively restricted to excitatory neurons, shown in blue, and absent from inhibitory neurons, again showing us then this distinction, this developing distinction between progenitors for the 2 major cell types in the cortex. And this particular clone is the one that I'd illustrated to you previously in blue. And there's another clade illustrated here in B. You can see that mutations B1, B2, and B3, as they become present in a smaller fraction of cells actually also become progressively more common in inhibitory neurons. So this is a progenitor that appears to be mostly, although not completely, specific for inhibitory neurons. And so then we can take these lineage marks and again, informatically plot them back on to the Teasney plot to determine what cell types the mutations occur in. So what you see on the left side is the Teasney plot now grayed out, but just showing the different islands. And then on top of it, you see cells that have particular DNA lineage marks. And you can see that the overall, the lineage marks pick up all of the major cell types all the major neuronal types in the cortex. You also see that the mapping is actually a bit better for the SmartSeq data set than the 10x data set because the SmartSeq is a larger data set and has richer transcriptomic richness. And then on the right side, you can see that intermediate to late mutations within clade C become limited to excitatory neurons and absent from the inhibitory neurons. And that the very last mutations in that clade then become limited to upper layer and middle layer excitatory neurons. So the paired-seq analysis of excitatory neurons in two different brains reveals an inside-out sequence of formation of neurons in the cortex. And so again, on the left in panel A, we see a variety of clades, labeled A, B, C, and D. And then for each one, you see mutations. And first, shown in red and blue, is whether the mutation is present in excitatory or inhibitory neurons. And you see then the graph with the dotted lines shows the fraction of excitatory neurons. And you see that basically then clade C in panel A becomes progressively enriched in excitatory neurons. And then on panel C, you can see for brain UMB 1465 above, or in panel D for UMB 4638 below, we've looked at a couple of those clades, and you can see that clade C from the first brain the later mutations become progressively restricted to upper and middle layer neurons in our SmartSeq data set and in our 10x data set. And then when you actually plot it onto the SmartSeq data set, the SmartSeq data set has amazing information about layer, because they micro-dissected the different layers. So again, you can see that the late mutations become progressively restricted to neurons in the middle and upper layers. And the same trend is visible in panel D for a couple of mutations from a second brain. And so this then demonstrates the inside-out sequence of birth date of neurons in the human cortex, where the earliest born neurons come to lie in layers 5 and 6, and the later born neurons are in the upper layers. And they contain mutations then that are not found in neurons of the lower layers because the lower layers become postmitotic earlier. So what we find then from this analysis, the neocortex is deeply polyclonal, that the neurons are born inside first, outside last. The upper layer neurons have mutations absent in the lower layer neurons. The progenitors become restricted to excitatory neurons, and we find that this occurs at a mutation mosaicism of about 4%. This allows us to estimate actually about how many excitatory neuron progenitors give rise to essentially one column of cortex, or the patch of cortex that we're analyzing. We estimate there's about at least 7, maybe 10, maybe a few more excitatory neuron progenitors that give rise to the excitatory neurons in any given patch. And that there's tremendous intermingling, so that neighboring neurons, shown in the cartoon in red or in blue or in green, actually derive from progenitors that are separated even all the way back to before gastrulation. And so that there's this intermingling of progenitors from vastly different progenitor types, even within a single column of the cortex. So the next question we can ask is, how does the output of a given progenitor distribute across the cortex spatially in relationship to the Brodmann areas? So this shows a flattened map of the human cortex. And this is a convention used by the Human Connectome Project. The numbers represent the Brodmann's areas, and my pointer again is not helping me, but there's area 17 way over on the right, shaded in yellow. There's area 4. You can see areas 1, 2, and 3 in light blue near the middle. That's the motor strip. Areas 4 and 5 are areas 1, 2, and 3 are the primary somatosensory cortex. Areas 4 is the motor strip in frontal cortex. And this is work of Sonia Kim, a graduate student, along with Sarah Bizotto, and using the data from Peng Peng and August. And so you can see then, she then took samples from various Brodmann areas, and an example is shown below that schematic in light blue. That represents a SNV that was discovered in single neurons of the prefrontal cortex, and then it shows its mosaic fraction in Brodmann areas 6, 8, 9, and 10. You see that this one clone distributes across these 4 Brodmann areas. And then we actually know the cell types that that clone gives rise to. We know that that clone is a very late lineage mark. It's almost completely restricted to excitatory neurons and in the middle and the upper layers of the cortex. So this is not even a clone that forms all of the layers of the cortex, and yet it distributes across multiple Brodmann areas in fewer than 1% of the cells. So a much broader pattern of distribution of this clone in a smaller fraction of cells than I think we were expecting to find. In fact, Sonia then focused her attention on the 17-18 border because it's such a sharp histological distinction. She sees some clones that respect that border. Here's a mosaic SNV, again, predominantly in excitatory neurons, as shown as in the bar graph on the right, and in this case limited to Broca's area 17 and not detectable in area 18 or 19 or in more rostral parts of the cortex. But here's a similar clone identified in area 17 in about 1% of the cells, but also present in area 18 in about 1% of the cells. And again, this is an excitatory neuron progenitor that, again, doesn't respect that particular border. We've done less so far with the lineage of inhibitory neurons because we don't have as much mutation information about mutations that mark inhibitory specific clones. This one clone I've shown you, we know distributes widely across the cortex, but the other mosaic marks we've analyzed so far are fairly early marks, and we have to analyze the cell types that they're found in. We see that they're found in all of the major inhibitory neuronal clusters, including somatostatin, parvalbumin cells, VIP, and so on. So we'll need to do more sequencing in order to get better marks. So finally, in the last few minutes, how do somatic mosaic mutations cause disease in the context of cortical development? Well, going back to the epilepsy mutations, we previously analyzed a handful of these lesions and identified what is the cell type that carries the MTOR mutation. We found that the smallest dysplasias show the mutation limited to new N positive neurons based on sorting new N positive cells from new N negative cells. And so these small mutations seem to be able to have epilepsy even when the mutation is limited to neurons. The larger mutations, not surprisingly, have the mutation in all different cell types. We think that, in fact, it's the excitatory neurons that are key based on mouse models where we created a mutation, an MTOR pathway mutation in PI3 kinase. We found that if you created that in the excitatory lineage using EMX1 Cre, you would create megalencephaly. If you did the same experiment in the inhibitory neurons using NKX1 2.1 Cre, in fact, you would not see megalencephaly. So we think that it's the excitatory neurons which are key. In fact, the size and shape of these focal cortical dysplasias resemble these excitatory neuron clones that I showed you. I'll skip this slide in the interest of time. So can we identify cell types carrying damaging autism mutations? Here is an example of a mosaic autism mutation that we identified in a brain that we think is causative. It's a mosaic CNV involving a portion of chromosome 2, and we know that germline duplications of this exact same region cause developmental delay and dysmorphic features. And so here is a very lucky event where in one of these 59 autism brains, we managed to capture a mosaic CNV event that we think is very likely contributing to risk because, again, it involves a known region known to contribute to risk. So we can see that it's a very complex duplication. It's actually a tandem duplication with an inverted duplication and an inversion. We developed PCR primers for several of these different breakpoints, and we could identify these breakpoints and show that they're present in new and positive neurons, as well as in new and negative cells, but they're definitely present in neurons in this particular case. It's present in about 20% of the sorted cortical neurons. And this just shows a very preliminary unpublished study by Caroline Diaz and Edward Strong in the lab, where they used RNA-seq to actually infer the presence of the CNV based on the RNA reads using a tool called infer CNV. And so at the top, you see the clusters of different cell types, the excitatory and the inhibitory neuronal clusters. And in the bottom, there's a zoom in on the excitatory and the inhibitory neuronal clusters. And I think the colors are not great, but the blue and the orange show that, in fact, it appears that the CNV is present in, again, about 20% of excitatory neurons and in about 20% of inhibitory cortical neurons. We'll need larger sample sizes to determine whether there's particular cell types that show particular enrichment of the CNV in the context of autism, or whether it's just present in relatively equal proportions of many different cell types. So just to summarize my talk then, the cell lineage and relative birth dates are inscribed in somatic mutations. There's this early divergence of excitatory inhibitory lineage that we can see in those mutations. We see that late-born neurons have additional mosaic mutations that are absent in the early-born neurons. We can see that the clonal structure of excitatory neurons doesn't so far show the kind of topographic constraints that had been suggested by mouse work. Although mouse studies with the same level of quantitative analysis have not really been performed, we don't know the mosaic fraction, for example, of those MADAM clones, so we don't know exactly how to compare apples with apples. FCD looks like it represents mTOR mutations in the excitatory lineage. It's an open question whether mosaic autism mutations show analogous cell type specific actions. Could mosaic mutations help us someday illuminate a cell type specificity of neuropsychiatric disorders? It's possible, but it's also possible that psychiatric disorders are fundamentally different in their functional organization. Perhaps mutations have to be present in multiple cell types in order to cause psychiatric disorders. In the interest of time, I'll skip this slide because I think we have to stop right on time, which just summarizes the advantages and disadvantages of mosaic mutations for lineage analysis. And I'll just conclude by acknowledging again the people who did the work. The mosaic autism work was done by Elaine and Rachel and Jan May, who were the leaders of it. And the cell lineage work using paired-seq, that was developed by Peng Peng in August. And Sonia Kim and Sarah have been doing the topographic work, and Sarah and Javier have been doing the early cell lineage studies. The mosaic CNV work was again done by Max Sherman in ProRu's lab, but collaboratively with our lab where we provided the sequence data, and Caroline and Rachel have been identifying the cell types. And there are many other former lab members that have contributed, and our lab has been fortunate to have a tremendous collaboration with Peter Park now for about 10 years, and there are many people in his lab that have enabled all of this work of single-cell mosaic mutation that would have been impossible otherwise. I just want to call out especially Max, who was formerly in Peter's lab before being in ProRu's lab, and Jan May Do. I think I've captured everyone, and I want to thank you all for your virtual attention, and I'm happy to take a few questions.
Thank you very much, I guess, from everyone. That was a fantastic talk and for sharing so many unpublished data or bioRxiv published data. There are some questions. I will go through some of them, the most voted one. So the first one is, are there somatic mutation hotspots in the genome that are commonly mutated in the population? And if so, are ASD regions enriched in such regions?
That's a great question. And so far, we haven't really seen particular hotspots. As I say, open chromatin is mutated at a significantly higher rate, but it's only a marginally higher rate, twofold higher or less. Again, exons are mutated at a slightly higher rate. We see the expected excess of mutations at CpG dinucleotides. So the development of mutations are often at CpG dinucleotides, and that's commonly seen in dividing cells in cancer as well. That seems to be due to the deamination of cytosine that occurs naturally so frequently, and then if it occurs in a cycling cell, that cytosine deamination event is then if it's replicated before it can be repaired, then it's converted into a permanent double-stranded mutation. And so we see those sort of common things, but we don't see anything that's unique to autism so far that hasn't been seen in cancer. There are recurrent mutations that cause these focal epilepsies, but we think that that's just not due to the fact that they are particularly mutable, but instead that they are particularly noisy. They're particularly disease-causing, and so are recovered at a very high rate.
So another question is, have you identified any extracortical affection by mosaic mutations related to ASD? I'm thinking in the thalamus. I'm thinking about the thalamus.
Right. We've been, that's another great question. I mean, we started by studying cortical DNA, and so we don't yet know the systematic distribution of the mutations we've identified throughout the whole brain, and we don't necessarily know what's the right structure in autism to focus our attention on. And so we haven't, to answer the specific question, we haven't so far found any particular mutagenic pattern, for example, in the thalamus or in any other part of the brain. We emphasize there's tremendous intermingling of progenitors within the brain. And so most mosaic mutations that we find, if they're present in more than about 1% of the cells, we can find them throughout the whole brain.
So another question from actually Stephanie Baulak. I just got it. Do you think brain somatic mutation may be present in circulating cell-free DNA and therefore be detectable not only in the brain but also in the biofluids?
Well, I think that it's fair to say that it can be detected in CSF. I know Michael Hildebrand in Australia has been doing some work that's not yet published on and analyzing CSF. We remarkably gave him one of our a mosaic double cortin mutation. He was able to detect in the CSF. I don't know whether they would be detectable in, microvesicles in the circulating blood or not. I think it's worth checking. I think anything's possible.
So Victor Borrell asked, do you think your clonal data indicates genesis of inhibitory neurons within the neocortex, siblings to excitatory neurons? So this would be different than mouse. Yeah,
so that's again a great question. As Victor knows, there's a couple of reports that have suggested that a subpopulation of inhibitory interneurons might be generated by progenitors in the cortical ventricular zone, whereas the vast majority are generated in the subcortical regions. I've tended to be skeptical of that claim over the years, but in fact, as you can see, as you probably saw from several of my figures, we consistently see in some of our clones a small number of inhibitory neurons in mutations that we would that are mostly excitatory restricted. At the moment, we can't be sure that this represents a dorsal origin of inhibitory interneurons. We can't rule out a technical problem, like, for example, two nuclei that got stuck together and that were interpreted as a single nucleus, so in other words, an inhibitory neuron that stuck onto an excitatory neuron. So we're not really certain yet. We'd like to, and also we haven't seen that these inhibitory neurons that appear to come from dorsal cortex have any distinctive transcriptional profile, and we'd feel more comfortable in our data if they were a particular subtype of inhibitory neurons. And so our markers are not yet rich enough to be able to define that subset. And so I think that question is uncertain, although I'm beginning to think it's more likely than not that there is a subpopulation of inhibitory interneurons generated from dorsal cortex.
I read a couple more, two questions. So one is, you mentioned that CMV known to cause ASD in germline state do not cause ASD in somatic state.
Right.
Does it imply that mutation needs to occur early in development, so in progenitor cells, for instance, to cause ASD?
By and large, that is true. So we'd expect that the Poru's haplotype phasing method can detect large CMVs even when they're in a tiny fraction of cells, as low as 1% of the cells. And yet most of the mosaic CNVs that we find as contributing risk to autism are in a large fraction of cells, have a fairly high mosaic fraction, 10 or 20% or higher. And so probably occurred in the first couple of cell divisions. And I think that may be something important about autism in contrast to epilepsy, but we're not really sure yet.
Next one is the different contribution of early cells to different neuronal types in the cortex reproducible between individuals, or does it vary? Is there a particular mechanism directing this, or is it a random process?
Yeah. So far we have limited data because it's very labor intensive to do all of these paired seq experiments. It's not a droplet-based method. So we only have analysis of a few thousand cells. And we've analyzed three individuals. We got fairly little data from one of the three. We have pretty good data from two individuals. They look broadly similar, but I would not say that we have enough data to be able to have the sensitivity to detect differences.
Maybe the very last one. Are mutations in chromatin histone modifiers enriched in ASD and related disease? Which is indeed is so. But have you looked into whether having mutations that affect chromatin accessibility could be associated with differential rates in somatic mutations?
Right. Yeah, that's a fascinating question. Yeah. So whether germline mutations that disrupt chromatin structure might leave the chromatin more open and might make it more highly mutable. I assume that that's what the questioner has in mind. And we haven't yet done that analysis. We've got the genome sequence, and so we also have germline mutations that are likely to be contributory to risk, but we haven't then gone back to ask whether those that carry germline mutations in chromatin have a higher rate of mosaic mutations. It's not obviously so far. So far the autism brains don't have a higher rate on average of mosaic mutations than the normal brains. But it could be that there's one case that has an outlier, and we have to go back and check. That's a very good thing that we should have done by now and haven't. Yeah.
Good. So there are several other questions, but I think we are now, it's 6 o'clock here in Vienna, and I think we have to thank you again, really, for participating to give this fantastic talk. And thank everyone for attending. We will have our next talk next Tuesday at the same time. So I hope to see, or not to see, but to hear from you numerous next time. So Chris, thank you very much.
All right, thanks. I want to thank all my friends who have listened and have been sending me little hellos along the way. And thanks again, Gaia, for doing this. It's a really wonderful thing to be able to do a little science while we're all stuck inside.
Thank you.
All right. Bye-bye, everybody.
Bye. Have a good day. Bye.
Yeah.